Alright, in this and the following two lessons we'll be studying the properties of DNA. DNA has three important properties that make it a hereditary molecule. Number one, it can be duplicated. And number two, it can store information. And number three, it can change. So in this lesson we'll take a look at DNA duplication. First, some background. We've already studied the cell cycle, composed of two main phases: interphase. And during interphase, the cell is burning sugar, it's making proteins, it's repairing, it's growing, etc. And then mitosis. Mitosis is when chromosomes are delivered into daughter cells. Now that means that during interphase, the chromosomes must be duplicated. And when chromosomes are being duplicated, that turns out that's DNA being duplicated itself, and we'll see that in a moment. We know that mitosis accounts for the increase in cell number during development. So we all start as a fertilized egg and then go through rounds and rounds of mitosis. The cells actually stick together to form the embryo and the body of the creature. Right? So mitosis accounts for development. We learned that mitosis itself consists of four stages. We call them the condensing phase and the lineup phase and the separation phase and the division phase. But remember, the chromosomes themselves get duplicated during interphase. And then mitosis is the process of delivering those duplicated sets of chromosomes to the two daughter cells. So it's time now to examine what a chromosome actually is. And in this diagram, complex diagram, shows a lot of information. To the right, or to the left here, we see a duplicated chromosome. Notice it looks sort of like an X. So here's one chromosome and here's its duplicate, but they're still attached. It's these two chromosomes that will be separated during the separation stage of mitosis. Now if we zoom in this little region of the chromosome, what is it? Well, see here, we see it's this supercoil, which then kind of unravels into smaller coils, and then we got these ball-like proteins called histones. We'll call them packaging proteins, and then we see this long molecule of DNA. This is the molecule called DNA, deoxyribonucleic acid, and notice its shape is sort of like a twisted ladder, or a double helix is what it's called. We'll think of it as a twisted ladder. Notice though how the DNA can be wrapped around these proteins, and that allows the cell to package a really long molecule of DNA into a small structure, a chromosome. And this only occurs during the condensing stage of mitosis. So when we say the chromosome is condensing, we mean that long molecule of DNA is being wrapped up into these structures, supercoils, so that then the cell can go ahead and deliver duplicates to the daughter cells. Now the DNA molecule itself has two backbones, right? So we got one backbone here and then the other backbone, so that would be like the sides of the ladder, and then the rungs of the ladder. This would be where you put your feet if you were getting up on the ladder. And you'll notice the way they're colored here, it seems as if there's a pattern. Each rung is made up of two colors, and those colors stand for certain kinds of chemicals that make up the rungs of the DNA ladder. So it looks like the green is always sort of paired with a yellow to make it rung, or a red with a purple. And that is true along the entire length of the molecule. Now the four chemicals that make up DNA have names, and they are as follows. Okay, so now we can get to the rungs of the DNA ladder. Here we see a model of DNA. Notice it's twisted still. And here we have the green and the orange, and then the red and the blue. The four chemicals, we'll call the orange one G for guanine, and the green one C for cytosine, and then we'll have the red one A for adenine, and the blue one T for thymine. And again, you'll notice that each rung is composed of either an AT combination or a GC combination. The red and white part here in this model would be the backbone, the deoxyribose part of the molecule, and we call these nucleic acids, the making up the rungs of the DNA ladder. But for us, we'll just remember the four letters. So G pairs with C and A pairs with T. And we say pair here because in fact there's a weak chemical bond here between these parts of the rungs. So the T is weakly bonded to A, which means it can separate right here at this center location here. Same with the G and the C. And that's going to be key to how this molecule is going to be duplicated. The first step in duplicating DNA then is that the two strands of DNA separate. So here we have still the twisted. Now we're showing that the two strands of DNA have separated. So those weak chemical bonds between the A's and the T's and the G's and C's have been broken by a protein, and now we have single strands of DNA. So the blue one is one strand of DNA, and here's the red one. Now convince yourself that the pairing rules, we'll call them the pairing rules, are still in force. So A pairs with T, the G was with the C was with the G, the C was with the G, the T was with the A was with the T. It's always those pairs. However, we've separated the two strands. Now notice then, if in the nucleus are spare parts, some isolated T's and A's and G's and C's, the rungs of DNA, if the cell has a ready supply of these chemicals, then when the two strands are exposed, the corresponding partners can match up with the chemicals along each strand of the DNA. So for example, we have here a C chemical floating around in the nucleus. If it bumps into a G, it'll stick. If the A finds a T, it'll stick. Notice here a T chemical bumps into the C, but they're not sticky with each other. Bumps into that C, it bounces off. Oh, but it finds its A and it sticks, right? So again, we have this concept of sort of stickiness. And this allows random collisions to actually create something that is ordered. So if you have a whole bunch of these individual chemicals and you have your two DNA strands exposed, then random collisions will allow these chemicals to find their matching sticky partners. And what you'll end up with then is two molecules of DNA, two identical molecules. So the DNA molecule has been duplicated. Convince yourself that these two double-stranded DNA molecules are duplicates. So we got GCTA, CG, 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 GCTA, CG, 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 etc. Right? So we originally had one DNA molecule, one double-stranded molecule. Each strand separates. Right? Then the DNA letters find their matching partners. There's a ready supply of these letters in the nucleus. And then the backbone of the molecule is stitched together, so that now it's a double-stranded molecule. Here's a sort of a textbook picture of that process. Again, here's the DNA molecule. It has unzipped, and here we see the two identical DNA molecules being produced. Now, what's not shown here are proteins that do some of this stitching work here. And again, we have to remember that these chemicals, the A's, G's, C's, and T's, are sort of randomly finding their partners, but then they get chemically bonded together to form the finished molecule. So one final point, remember, DNA duplication is only going to happen if the cell is going to go through mitosis, right? If the cell is going to go through mitosis, it has to duplicate its DNA so that daughter cells can receive the full set of chromosomes. But let's imagine these are the cells of the adult body. Well, those cells maybe are not going to be dividing anytime soon. They're still going to be utilizing their DNA. And to understand that, let's go back to this picture here. The idea here is that DNA is now, we're going to learn, has this second important property. It can store information. And when the DNA is exposed like this, the cell can access the information of DNA. What is this information? We'll learn that it is the recipe to build a protein. And one chromosome would have many recipes to build many proteins. That's why it's so long. It may have the recipes to build hundreds, even 1,000 or so different proteins in one chromosome. But in order to access that information, the DNA has to be accessible, right? During mitosis, when the DNA gets all wrapped up, then the cell is sort of wrapping up the DNA, and the DNA is not accessible. The information is not accessible. But that's okay, because the point of mitosis is simply to deliver the chromosomes to daughter cells. Then, during interphase, when the new cells, daughter cells, are produced, the chromosomes unwind again so that the DNA can be accessible, so its information can be used for cell activities.